All right, let's talk about one of the coolest, most powerful tools in all of modern medicine. I'm talking about monoclonal antibodies. And these aren't your typical drugs. Nope. They are literally engineered versions of your body's own special forces designed to hunt down disease with unbelievable precision. To really get it, you've got to appreciate that your body, right now, is basically a battlefield. Seriously. Every single day, you're getting bombarded by viruses, bacteria, all kinds of invaders. It is a constant microscopic war. And in that war, your immune system has its own elite soldiers. These are the snipers, the special ops unit, and they're called antibodies. They are absolutely the key to winning the fight. So here's our game plan. We'll start with how your body naturally creates these defenders. Then we'll see how scientists basically captured that magic to build a perfect antibody, and we'll look at the anatomy of these so-called smart bombs. After that, we'll follow their journey from mice to medicine, see how they're fighting disease today, and then we'll take a quick peek at what's next. Okay, first things first. Let's look at what's happening inside you naturally. Your body has this incredible, almost magical ability to create an army of antibodies with what seems like an infinite variety of weapons, one for pretty much any enemy it could ever face. So how do they actually work? Well, think of them like perfectly crafted keys. They're designed to lock onto very specific molecules on an invader, we call those antigens, and they do it with pinpoint accuracy. You've got millions of different cells making these antibodies, each one making a unique key for a unique lock. This, by the way, is the whole secret behind immunity. Your body remembers which key worked and keeps it on hand for next time. Which brings us to this amazing biological puzzle. I mean, think about it. You don't have nearly enough genes to have a separate blueprint for every single one of these millions of antibodies. So how on earth does your body pull off this incredible feat of diversity? The answer is kind of like a genetic Lego set. Instead of one huge fixed blueprint, your cells have a library of different DNA blocks. An enzyme comes along and acts like molecular scissors, making a totally random cut to choose which blocks it's going to use. And that cut isn't always perfect. Sometimes the repair process creates these little unique, kind of frayed edges on the DNA. This just adds another little sprinkle of randomness to the final design of the antibody. And then it gets even wilder. Another enzyme just shows up and starts adding brand new, completely random DNA letters to the ends. It's not copying anything. It is pure genetic improvisation. It's just making stuff up, which guarantees every result is gonna be unique. And finally, those two customized ends get stitched together. And what you're left with is a totally unique coding joint. So this whole process, shuffling, cutting, improvising, creates a brand new gene for a one-of-a-kind antibody. It's amazing. So if the body is this incredible antibody-making machine, the next huge challenge for medicine was figuring out, okay, how do we capture that lightning in a bottle? This was the billion-dollar question. How do you find that 1 in a million cell that's making the absolute perfect antibody against, say, a cancer cell? And then, how do you make billions and billions of identical copies of it? Well, the big breakthrough, the game changer, came in 1975. Two scientists, Kohler and Milstein, came up with something called hybridoma technology. And yeah, they won a Nobel Prize for it. From there, it was decades of refining the process, learning how to mix and match mouse and human parts to create what we now call chimeric and humanized antibodies. The hybridoma process itself is just a brilliant piece of bioengineering. First, you inject a mouse with whatever you want to target. The mouse's immune system does all the hard work and creates the perfect B cell for the job. Then, you take those B cells and you fuse them with immortal cancer cells called myeloma cells. The result? A hybridoma. It's a hybrid cell that has become an immortal little factory, just pumping out that one perfect antibody forever. All right, so now we have our factory. Let's take a closer look at the actual weapon it's producing. What is it about this Y-shaped molecule that makes it such a powerful medical smart bomb? So the key word here is monoclonal. It means they all come from a single clone of cells. That means every single antibody in the drug is a perfect identical twin. They are an army of clones, all programmed to hit the exact same tiny spot on their target. And that is what gives them their surgical precision. Now that Y shape has two really important parts. The arms are called the Fab region. Just think A and B for antigen binding. This is the part that changes to recognize millions of enemies. The stem is the FC region, and here's a great way to remember what it does. Just think of the letter C. It's the constant region. It's where the complement system gets activated. It's where other immune cells can bind. And it controls the antibody's class or isotype. All Cs. So we had the factories, we understood the anatomy. But there was one pretty big problem. Remember how this all started? With a mouse. And our immune systems are really, really good at spotting and attacking foreign proteins from a mouse. The solution was to humanize the antibody. It's genius, really. Scientists used genetic engineering to basically snip off the super specific antigen-binding tips from the mouse antibody and then graft them onto a human antibody backbone. They swapped out the mouse's constant region for a human one, creating a hybrid or chimeric molecule that our bodies are way less likely to attack. That made the therapy so much safer and more effective. And with that problem solved, the whole arsenal got a massive upgrade. The doors were basically thrown wide open for a whole new era of targeted therapy, letting us wage a modern war on some of our most devastating diseases. And their use cases are just incredibly diverse. For cancer, they can be designed to stick only to tumor cells, basically painting a target on them for the immune system. Or they can block signals that tell the cancer to grow. For autoimmune diseases like rheumatoid arthritis, they can find and neutralize the specific molecules that are causing all the inflammation. And for infections, they can literally grab onto a virus, like RSV or the one that causes COVID-19, and physically stop it from getting into our cells. And this isn't some small niche part of medicine we're talking about. The global market for these therapies is projected to hit $300 billion by 2025. That number tells you everything. This is a fundamental shift in how we treat disease. But you know what? This revolution is still happening. It's unfolding right now, and the future is all about getting even more targeted, even more precise, and even more clever with our designs. You see, today we're already moving beyond the hybridoma method. With amazing gene editing tools like CRISPR, scientists can now design the perfect antibody from scratch, entirely in a computer and a lab. This gives us a level of control that we've just never had before. And that power leaves us with one final huge question. With this ability to build the perfect biological weapon against disease, what should we target next?